The first 1,000 days of life have long been recognized as a critical window for brain development, immune maturation, and metabolic programming, but emerging research now suggests this period may also lay the foundations for how we experience hunger, satiety, and food preference for the rest of our lives. At the center of this developmental process is the early life microbiome, the community of bacteria, viruses, fungi, and other microorganisms that begin colonizing the infant gut from birth. These microbes don't just digest milk or help ward off pathogens, they shape the architecture of the gut-brain axis, forging neural circuits that may determine whether we grow into intuitive eaters, chronic overeaters, or adults with disordered relationships with food. At birth, the human gut is essentially a blank slate, nearly sterile and immunologically naive. Within hours, however, a flood of microbial life enters the newborn's digestive system, depending heavily on the method of delivery: vaginal birth versus C-section, feeding, breast milk versus formula, antibiotic exposure, and maternal health. This first wave of microbial colonizers is not random. It plays a pivotal role in educating the immune system, training the intestinal barrier, and setting in motion the production of key neurotransmitters and signaling molecules. These early microbes help establish the gut's neuropod network, enabling the digestive tract to act as a sensory and communicative organ that reports directly to the brain. It's during this developmental window that the gut learns to read the chemical landscape of food. Specific microbes ferment carbohydrates into short-chain fatty acids, SCFAs, like butyrate and propionate, which in turn influence gene expression in gut epithelial cells and modulate appetite-regulating hormones like GLP-1 and PYY. These hormones don't just suppress hunger, they also shape reward perception and the brain's valuation of different foods. In animal models, germ-free mice, born without gut bacteria, exhibit abnormal expression of these appetite regulators, underdeveloped vagal circuits, and impaired satiety sensing. When microbes are introduced early in life, some of these pathways can be normalized, but the window for full recovery narrows with age. This microbial education extends beyond caloric regulation to taste preference itself. Infants exposed to a diverse range of flavors through breast milk, which reflects the mother's diet and microbiota, tend to grow into toddlers with broader palates and greater acceptance of fruits, vegetables, and fiber-rich foods. Conversely, a lack of microbial diversity, often caused by early antibiotics, poor maternal nutrition, or formula feeding, has been linked to an increased preference for sweet, salty, and high-fat foods. This may reflect altered microbial production of dopamine precursors and disrupted signaling in the brain's reward centers, effectively hardwiring the brain to seek calorie-dense foods even in the absence of need. The long-term implications are striking. Several cohort studies have now linked early life dysbiosis to increased risk of childhood obesity, type 2 diabetes, and even anxiety or depression, all of which influence eating behavior. One study found that infants with low levels of bifidobacteria and lactobacillus at 6 months had higher BMI and altered eating patterns by age 7. Another observed that certain microbial patterns in infancy predicted greater responsiveness to food cues, emotional eating, and food fussiness years later. These aren't just correlations, they suggest a causal role of gut microbiota in shaping lifelong neural circuits related to food. Even brain imaging studies now show that the composition of the gut microbiome in early life is associated with changes in the volume and connectivity of brain regions involved in interoception, impulse control, and reward regulation. This includes the insula, amygdala, and orbitofrontal cortex, areas that govern our ability to detect hunger and fullness, resist temptation, and find satisfaction in eating. A gut microbiome skewed toward inflammation or lacking in microbial metabolites like GABA, serotonin, and SCFAs may leave these systems underdeveloped or hypersensitive, creating a brain that is chronically uncertain about when and how to eat. Importantly, this is not deterministic. While early microbiome development exerts a powerful influence, it remains modifiable through diet, environment, and targeted interventions. Breastfeeding, diverse whole food diets in early childhood, and minimizing unnecessary antibiotics can help foster a robust gut ecosystem. In high-risk infants, next-generation probiotics and prebiotics tailored to promote neuroactive metabolite production are being explored as tools to correct developmental trajectories. Moreover, understanding these microbial roots may help explain why some individuals struggle more with self-regulation around food, not because of weak willpower, but because their biological programming was altered from the start. In sum, Early life microbiome development is not merely a digestive milestone, it is a neural and behavioral blueprint for how we experience hunger, satiety, and pleasure from food. As science deepens our understanding of this hidden sensory axis, new doors open for preventative strategies that support healthy eating behavior from infancy through adulthood. The microbiome, it turns out, may be the first and most enduring voice in our lifelong conversation with food.